Hi, we continue our discussion of proteins translation, and we will look at the ribosome in detail. So the ribosome, the complete one here, is a very large protein complex, 4.2 megadaltons in size. It has two major subunits, the large subunit and small subunit. The sizes are characterized by their sedimentation rate. As you can see, it's not a linear property. S stands for Svedbergs, by the way. The large subunit is comprised of approximately 49 proteins and 3 ribosomal RNA molecules. The small subunit is made up of 33 proteins and a single RNA molecule. And the mass contribution to the final weight is approximately equal between the protein subunits and the ribosomal RNA molecules. Ribosomes themselves are some of the most abundant proteins in a cell. In bacteria, it can represent 2 to 3% of the total protein mass, so the cell is always geared up for making a lot of them. In eukaryotes, they're made in the special part of the nucleus called the nucleolus. The nucleolus is depicted here in blue, and the nucleus is surrounded by a yellow membrane. The genes for the proteins and for the ribosomal RNA exist on nearly every one of your 23 chromosomes, and parts of every chromosome loop into the particular nucleolus region, where there is a lot of synthesis of the genes for the ribosomal RNAs and the ribosomal proteins. Now the messenger RNA for the ribosomal proteins will get transported out into the cytosol. There, the messenger RNA will be turned into ribosomal proteins by existing ribosomes. Then the individual ribosomal subunits will be transported back to the nucleus, and subunits will be assembled into larger complexes inside the nucleus, and finally exported as intact large and small subunits in order to form more ribosomes in the cytosol. The only reason telomerase is included in this depiction is because it's also a complex of protein and RNA, and it is also assembled in the nucleus, but it's not pertinent to the translation story, however. On this slide, we see 2 of the 3 rRNA molecules, ribosomal RNA molecules, all wound up from the large subunit. And we also see, just for reference and size, 1 of the 49 ribosomal proteins in the large subunit, that being L1. So if all the proteins were present and we drew everything to scale, the final size of the ribosome complex would not be that significantly different from what you see here. Now we tend to think mostly of proteins doing biochemistry, but in the ribosome the RNA is very much a participant of the machinery that's actually doing the chemistry. It's not just a structural role here, and that's why the ribosome can be referred to as a ribozyme. Here we see a picture of large subunits and the small subunit and 3 transfer RNA molecules. Again, no proteins are present here in this picture. And as the diagram in the middle shows, that the messenger RNA that is being decoded will run through the ribosome approximately right here. Now there are 3 places or special sites for tRNAs in the ribosome, and over here there are 3 tRNAs sitting in those sites. The sites are the exit site, or E site, the peptidyl site, or P site, and the aminoacyl site, or A site. At the peptidyl site, this is where the tRNA sits that has the peptide attached to it. The aminoacyl site is where the tRNA that's bringing the new amino acid is connected. You will need to memorize the names of these sites. Now here we can see the 4 steps of one cycle of peptide elongation. In the peptidyl site, the P site, you have an existing polypeptide chain attached to a single tRNA molecule. The correctly base paired tRNA, the one with the proper anticodon for the existing codon, comes into the A site, the aminoacyl site. And the existing amino acid will attack the carbonyl end here, cleaving the bond between the amino acid and the existing tRNA in the P site. And now it will be attached to the new tRNA. The large subunit will now shift 1 codon, and then later the small subunit will shift 1 codon, and then you are back at step 4. Once the tRNA is left, you're back in the same situation as we were in step 1. This is a fairly straightforward description. And here is a very short YouTube video, again about 2 minutes, that really does a great job of showing it in animation. So I recommend that you look at that. In this slide, we'll talk about how the ribosome takes the initial steps of protein synthesis. Here we see a mature mRNA molecule. And just as a reminder, the beginning of the mRNA molecule is started from the transcription start site. All proteins start with the amino acid methionine. It's kind of a random fact, but that's just the way it is. So this is where the translation begins, the translation start site. Since we saw in the previous video that there's only one way to code for the amino acid methionine, the translation start site will always be represented with the nucleotides A, U, and G. And it's the tRNA for methionine in conjunction with the small ribosomal subunit that will bind to the mRNA, and it will start to scan for the AUG sequence where translation will start. It's a current puzzle that it's not always the first AUG that will be encountered that will be the translation start site. Sometimes the complex only starts on the second or third AUG, and that's still a question of active inquiry that we don't understand, how the ribosome figures out which is the correct AUG for starting. There's also a sequence usually in this region where the ribosome is likely to first bind, and that's sometimes referred to as the Shine-Dalgarno sequence. Once the small subunit finds the AUG start translation codon, the large subunit will come on, and now you will assemble a full intact ribosome on the RNA, and protein synthesis will proceed. Now we saw that bacteria have polycistronic mRNAs, RNAs which cover an entire operon, and from which multiple proteins that have connected function can be synthesized from. Pictured here are 3 Shine-Dalgarno ribosome binding sites, in this case one for each of the 3 proteins. And the final slide in our discussion of translation is how the process of translation ends at stop codons. These 3 stop codons are recognized differently from the codons that code for amino acids. They are not recognized by a tRNA molecule. Rather, they are recognized by a protein that binds to these 3 sequences. This is the signal to release the newly synthesized peptide, disassemble the ribosomal subunits, and release the mRNA molecule, allowing it to bind to another ribosome to synthesize another copy of the protein. This is the shape of the tRNA molecule, and this is the shape of the stop factor protein. As you can see, there's a fair similarity here in the overall shapes, which is not surprising since they have to fit into the same A site on the ribosome. But it's pretty interesting to see that the same shapes can be achieved by these very different types of molecules. Alright, in these 2 videos on translation we have covered a lot of ground. We've talked about the mRNA reading frame, how codons specify for amino acids, the roles of tRNA molecules, the direction of protein synthesis from the amino end to the carboxy end, and the 4-step cycle of peptide elongation. Thanks for listening.